Welcome to the second half of our course material. Yep, we will be covering 20 chapters. Also, with Chapter 11, we will kick off Unit 3, where we discuss membranes, the processes that rely on, and the processes that rely on them. Chapter 11 will focus on membrane structure. Here are our objectives for this chapter. As always, I expect you to have these mastered by the time we take our next exam. This may be entirely new material for you, so if you are struggling, please let me know so we can sit down and work through these concepts. Here is an outline for this lecture. We will be talking about the importance of membranes, osmosis, the properties of the lipid bilayers, membrane proteins, membrane carbohydrates, and we will wrap up the lecture with a short discussion of how cells control the cell membrane. Before we get too carried away with membrane structure, let's review a few concepts about membranes. The first of these is that the membrane is the basic barrier between the intercellular components and the rest of the cell, as well as a barrier between the cell and its environment. We call the membrane that surrounds the cell the plasma membrane. All membranes in the cells are made of a similar structure of two layers of phospholipids. The remaining components of the membranes can vary depending on their purpose. We will frequently refer to the membranes as a fluid mosaic because all the components of the membrane move fluidly and contain a variety of components that are kind of like the small bits of tile in a mosaic. Regardless of their specialization with regards to their function, all membranes serve the cell as a selective barrier, only allowing certain particles to pass through without problem and keeping the rest out of the inner area or forcing these molecules to use special transport proteins. Overall, while we focus on a lot of the other components of the cell, without these membranes, the cell would cease to exist. One more quick reminder on membranes has to do with the flow of water in and out of cells. This brings up our old friend, osmosis. Let's review osmosis real quick. Water will always move down its concentration gradient, with the goal of making the concentration of water in solute equal on both sides of the cells. This is why physiological saline is so important. If we were to place a cell in pure water, and because water can pass freely through the membrane, the water outside the cell would rush into the cell. This would cause our cells to rupture, unless they have a cell wall, in which case the cell has normal pressure. The converse is also true. If we were to place the cell into a solution with a higher salt concentration than inside the cell, the water inside the cell will pass through the membrane and exit the cell. This causes the cell to shrivel up. I've posted some videos of this on Blackboard so you can see the impact of water leaving the cell and coming back into the cell. We aren't too worried about osmosis right now. The gradients are very important, so make sure you understand the basics of gradients and how this impacts molecules. The primary component of any membrane is the lipid bilayer. This is a universal component of all cell membranes, regardless of size or function. In this section of the lecture, we are going to explore what the bilayer is, how it is formed, some of the properties of the lipid bilayer, and finish up by briefly discussing how membranes are made. Hopefully you remember from the beginning of the semester when we talked about the macromolecules of the cell. If not, I suggest you review the chemistry chapter. In any case, when we discussed lipids, we talked about a specialized type of lipid known as the phospholipid. The phospholipid is constructed of primarily two parts. The first of these is the head, which is the hydrophilic or water-loving part of the molecule. The head of the phospholipid is made up of three parts: a choline moiety, a phosphate group, and a glycerol. We usually represent this head as a little oval. But as you can see, this is a simplified view of the head. The tail is more basic. It consists of two fatty acids. In general, one is a saturated fatty acid, while the other is an unsaturated fatty acid, giving it the kink that you see in the diagram. This is simply the most common form of phospholipid. There are other structures that are found, but as far as we are concerned, this is the primary phospholipid. This molecule highlights a common quality that all components of the membrane have. This is known as amphipathic. The molecules that are known as amphipathic have regions that are hydrophobic and hydrophilic. The lipid bilayer is made of two rows of these phospholipids. The tails of both sides of the lipid are on the inner side, while the heads form the outer edge of the membrane. This structure is not a random formation, but is driven by the properties of these molecules. When exposed to water, the hydrophobic ends of the molecules will naturally group together due to being excluded by the water. This leaves the heads of the molecule left on the outside of the structure, exposed to the water. Due to this behavior, membranes have two pretty cool properties. The first is that they self-seal the membrane. This means that if a phospholipid is damaged or a puncture has occurred within the bilayer, the phospholipids will move around in such a way as to repair the damage. The other cool thing that these bilayers can do is to spontaneously form sealed compartments. This is how vesicles are formed within the cells. We use this behavior as a part of drug delivery mechanisms. Another aspect that comes about due to the nature of this bilayer is the fluidity of the bilayer. The bilayer is always in a state of motion. The phospholipids are always switching places or moving around within their spots. And the proteins and other membrane components can move around. This creates quite a unique environment that can allow for a great amount of activities. One example is how a cell surface receptor can dimerize to begin a chain reaction within the cell. The amount of fluidity is dependent on its composition. As we have already touched upon, the main component of the membrane is the phospholipid. It would then make sense that this can have an important impact on the fluidity of the membrane. There are two ways that these phospholipids can change the viscosity of the membrane: through the length and the type of fatty acids used in their tails. As we already know, there are two types of fats: saturated fats and unsaturated. And from a few slides ago, you should remember that the common phospholipids have one of each. If the composition of the membrane includes more unsaturated fatty acids, the membrane will be more fluid, and you will see components moving around more. Whereas a membrane with more saturated tails will be more viscous, and the components of the cell will not move within the membrane as readily. Besides the amount of double bonds in a fatty acid, the length of the tail can have a similar impact. The range in tail length can be around 14 to 24 carbons in length, with the majority around 18 to 20. The shorter the tail, the more fluid the membrane can be. Why does membrane fluidity matter? It helps us control the rate of interactions, and as you can tell when you forget to put the butter back in the fridge, temperature has a direct impact on the lipids. By swapping out the phospholipids for ones that maintain the desired fluidity at the temperature the cell is at, the cell can maintain functions in temperatures outside of its normal range. This is especially true for single-celled organisms. Eukaryotes take this one step further by introducing cholesterol to the bilayer to help maintain the fluidity. We will talk more about cholesterol at the end of this chapter. The big take-home message here is that in order for cell membranes to participate in their three main functions of receiving information, importing and exporting molecules, as well as movement or expansion, the membrane must be fluid enough to allow for these changes. This all adds up to fluidity being one of the most important aspects of cell membranes. It shouldn't come as a surprise to you that given the nature of the cell, there is some imbalance in the way the cell membrane is set up. There are specific components that are found on the outside of the membrane and certain components that are required to be on the inside of the membrane. And even if they are the same components, they may be found in different quantities and in different places. We are going to talk more about this asymmetry when we discuss the other components of the membrane in a few sections. Membranes do not come into being magically. They are produced by the cell, specifically by the endoplasmic reticulum. I'm sure somewhere in your memory is a discussion of how the ER is responsible for transportation to the surface, and maybe you remember a picture of a vesicle budding off and going to the magic place where vesicles go. While this is the basics, it is much more than this. The membrane is formed by the ER, which synthesizes phospholipids, specifically the smooth ER. These phospholipids are generated in an uneven way. As you can see from the image, the one side gets the phospholipids added to it unevenly. So how are these how are these bilayers evened out? There is an enzyme known as flippase. Yeah, I know, it sounds like I made it up. That helps transfer these phospholipids to the other side of the bilayer. So now you know how the major component of the membrane is formed, but what about all the other components? The cell maintains all membranes in the same way. This is known as preservation of orientation. In short, the cell regulates membranes by always having a cytosolic phase and a non-cytosolic phase. For organelles, this means that the non-cytosolic phase is the internal side of the membrane, while for the plasma membrane, the non-cytosolic phase is on the outside of the cell. As the components of the cell membrane are prepared in packages, proteins meant to be on the extracellular side of the cell are kept on the internal sides of the vesicle. And the same holds true for plasma membrane components that face the inside of the cell. When being formed in vesicles, they face outside the vesicles. Once production is completed, the vesicle will pinch off and go where it is destined, either to another organelle or to the cell surface. Once it has arrived at its destination, the vesicle will fuse with other membranes and join the existing structure. That's it for this part of the topic. We'll come back and cover the rest of Chapter 11 in Topic 2.